a seminar series after a break of a couple of months. We have Ashmita today talking about CE1, and we're really excited to see all the work that that team has been doing. And then we'll take a little break with the seminar series for the summer. And we'll be back in September, October, and November for presentations from Carlene and team with CE2, the social science team in October, and on-farm team talks in November. So we're looking forward to that as well. We've got great attendance today already, although I assume that over the next couple of minutes, people will continue to filter in. But I'm really excited to have Ashmita joining us. Ashmita is a postdoc with Hannah at University of Kentucky, and I just learned that she's actually on the ground in Wisconsin, in Madison. So, you know, it's really great to be at the point in this project where we're starting to get these lessons learned across the project. So I'm excited to have you share them. So Ashmita, I'm going to let you take it over from here.
Thank you, Jen. Right, let me share my slides quickly. Okay, do you all see my slides?
Yes, we do. Looks good.
Thank you for the introduction. Yes, as Jen mentioned, I am a postdoc with Dr. Hannah Poffenboscher at the University of Kentucky. And today we're going to share our learnings from Common Experiment 1, and I've titled this talk as Cover Crops' Effect on Nitrogen Cycling and Crop Yield. So as we all know that after Industrial Revolution, 1950s, there was a bloom in nitrogen fertilizer use, and that kind of helped prompt agriculture revolution as well. So we need nitrogen fertilizer to feed the growing population, but at the same time, under and over application of nitrogen may lead to decreased yield and environmental ramifications. So nitrogen can be taken up by plants in two ionic form, ammonium and nitrate being a negatively charged ion, can get leached down from the soil profile and can cause groundwater pollution and can also cause hypoxia in water bodies. Similarly, nitrogen can get lost from soil in other forms like gaseous form through ammonium volatilization or nitrous oxide emission as well. And this is the data from the year 2022, and USA here ranks third among in the world in nitrous oxide emission, and agriculture sector apparently contributes the most to the nitrous oxide emission in the US, followed by industry, waste, land use change, etc. And the largest emitter of nitrous oxide in agriculture are the synthetic fertilizers, followed by manure mismanagement. Therefore, it is important to optimize nitrogen fertilization in agriculture. And among major food and fiber crops in the US, corn dominates the major food crop. And these are the numbers that I got from USDA 2020 in the year 2023, and they represent area harvested in 2023 in million acres. And on your right, you're looking at a map that shows corn production in the US, and darker green color represents geographical locations with which contains major corn producing regions, and lighter color represents intermediate to minor corn producing regions. And it includes states like Illinois, Nebraska, Iowa, Wisconsin, Kansas, Indiana, Minnesota, and these are also in our, most of these sites are in our in our study locations as well. So our study locations include Illinois, Nebraska, Indiana, Kansas, Wisconsin as our, and they are also major corn producing states in the US, and then Kentucky, which is in the intermediate side. And then we have Pennsylvania, North Carolina, New York, Maryland, and Vermont. And these numbers, they show area harvested for grain in the year 2018, and I also got it from USDA-NAS website. So we have already established that it is kind of important to optimize nitrogen fertilization in corn cropping system, but it is challenging at a field level due to the complex genotype management and environmental interactions, which include genetic variations, weather variations, climatic variations, soil heterogeneity, disease pressure, also different residue management strategies, fertilizer management, different cropping systems, etc. Cover crops, crop protection, etc. So one of the ways of optimizing nitrogen rate is by determining N rate based on yield in response to nitrogen fertilizer rate. And we did do that by fitting in a quadratic plateau response curve to yield in response to nitrogen fertilization, and that can give us agronomic optimum nitrogen rate when you and that agronomic optimum nitrogen rate is the break point between this quadratic and plateau curve. And that and what it means is by adding more nitrogen beyond this point, there is no substantial significant increase in the yield. And then this slope represents increase in yield per unit nitrogen input, and the intercept represents the yield at no nitrogen, that is zero nitrogen fertilizer nitrogen input, and that indicates a nitrogen supplying capacity of soil and residue. And another way is by determining economic optimum nitrogen rate, which is normally less than agronomic optimum nitrogen rate because it takes into account price associated with nitrogen fertilizer as well as corn grain price. And what it means is by adding more nitrogen beyond economic optimum nitrogen rate, you might be able to increase yield slightly, but that is not economically viable option for the farmers. And this also takes into account the surplus nitrogen that is available to the crop from previous crop, to our corn from previous crop, from soil, from cover crops, but that can get lost from soil via either leaching or in the gaseous form through denitrification or volatilization. And one of the ways to retain it is by planting cover crops. So the concept behind it is the cover crops, as the name suggests, they can cover the ground, hence protecting soil loss from extreme weather events, but also at the same time protecting nitrogen from that soil. And other idea is that the nitrogen that is in the soil, the surplus nitrogen that can be retained by the cover crop in their biomass via assimilation and through decomposition based on carbon to nitrogen ratio of that biomass that can become available to the following crop. At the same time, they can, based on the type of cover crops, they can also capture nitrogen from the atmosphere. But there is uncertainty in around the optimum nitrogen management following, and that can decrease or increase, that can change the optimum nitrogen requirement for the corn because of that nitrogen that might become available to the to corn after we from the cover crops. But because the uncertainty is there because it depends on lots of factors here, as I described, like cover crop species itself, but then there is soil, physical, chemical, biological properties, texture, moisture, temperature, soil microbes, diversity, biomass of microbes, and then there's weather condition, temperature, precipitation, and there is residue quality and management, termination timing. And some of the I'm going to present some of these contradictory experiences and perspectives around cover crops, advantage to the next crop. So some research have found that can adding cover crop, planting cover crops, they can cut fertilizer costs for the next cash crop, and the idea is same by scavenging or adding nitrogen to their biomass and then making it available to the next crop, thus cutting fertilizer cost. And in some surveys, it was found that around half of the people, respondents in the survey, they actually reported that they applied extra nitrogen around the time of corn planting when following winter cover crops, and half of them were like they didn't have to do that. And in some of the research, it was like after cereal rye, after the grass residue, there was nitrogen was not really available to the corn because the sequestered nitrogen was not available at the time when corn actually needed. So therefore, this can, because of the uncertainty and contradictions, all of that, this can complicate nitrogen fertilizer management for the subsequent corn. And for that, the objective of our study is, therefore our study is, we have used this multi-site year field experiments where we have 12 corn sites, as I mentioned before, and then this experiment was conducted for 3 years. And we used multiple winter cover crops, different species, legume, rye itself, and then mixture of legume and rye to obtain this better understanding of how winter cover crops can affect the optimum nitrogen rate of corn. And our hypothesis was that legume cover crop will decrease the optimum nitrogen rate requirement for corn, whereas cereal rye will increase it compared to legume and or fallow. And mixture will be in between legume and rye, given its mixed components of legume and rye. And the concept behind it, idea behind it, is that legume with a lower carbon to nitrogen ratio might, and also their ability to capture nitrogen directly from the atmosphere, might be able to provide more nitrogen to the next crop, or it will decompose faster. So higher and faster decomposition might decrease the relative optimum nitrogen rate for corn compared to rye. So for that, so I already mentioned we had 12 sites, and there were 3 years, and this was the design was randomized complete block split plot design, where our main plot was cover crops split into nitrogen fertilizer rates, and we had 6 nitrogen fertilizer rates starting from 0 to 320 pounds of nitrogen per acre, and there were 4 replications per site here. And all our sites were managed under no-till, and most did not have a history of cover crop use, but they all almost had did annual crop rotation, and the previous cash crops were silage or grain corn, winter wheat, soybean, oats, and barley. Yes, and the legume species, there are 2 different legume species that were used in our sites. For most of our northern sites, they use hairy vetch, and southern sites use crimson clover. And beside nitrogen, all the fields were unlimited by other essential nutrients like P's and K's. Little more description about our site. So I've used this principal, done this principal component analysis, and to describe, and the variables that I used were our static soil variables like texture, sandsill clay percentage, organic nitrogen, available water storage, and 30-year climate normals like temperature and precipitation. And our PC1 and PC2 were able to describe or explain 75% of the variability in our data set. And here, the red words are the variable names in red, and then the blue lines, they represent where the gradients are, and the sites are over here. So I'll explain that. So Nebraska, Kansas, Wisconsin, they're more towards like they have clay and silt percentage higher in their soil, whereas Delaware had more like sandy soil. And Maryland, more site with more precipitation, whereas Wisconsin with higher towards more positive organic nitrogen gradient, so it had more organic nitrogen in their soil, and North Carolina, it was higher temperature gradient, so they had warmer temperature compared to Vermont, Wisconsin, New York, Pennsylvania. They were towards more like cooler. They were cooler sites, so towards lower temperature gradient, and Kansas had more available water storage. It is the volume of water in the soil that becomes available to the plant when soil reaches its field capacity. And then Illinois, Kentucky more towards like intermediate in the temperature reason, and then Indiana more towards like intermediate in the available water storage. And I've also wanted to show this daily average precipitation and temperature for our locations. This is for now for just the three our years, experimental years. And this precipitation range from 1.7 millimeter to about 3.5. And temperature range from 9.9 to 17 degrees Celsius. And all 3 years were almost similar in terms of their daily average precipitation and daily average air temperature. So now these three colors represent three different experimental years. And this shows a timeline of cover crop planting and termination. And we have arranged our sites in such a way that based on latitude. So higher or northern sites are on the top, and then southern sites are towards the bottom. So here cover crop planting kind of range from September to mid-October, where like you might notice Vermont, Nebraska, Kansas sort of planted their cover crops in September, whereas sites like North Carolina, Delaware, Maryland sort of planted them later in the October, mid-October. And this was similar in all 3 years. And in case of termination, cover crop termination, which was done chemically, and it was some sites did it like 5 days before planting the subsequent corn, and some even waited till for a month. And New York, Pennsylvania kind of terminated their cover crop a little bit late in mid-May, whereas as compared to other states. And then after cover crops were terminated, corn was planted either in May and it also ranged from May to June. Some sites like Kansas, Delaware, North Carolina did it earlier in May, and some as compared to other sites like New York, Pennsylvania, Maryland, and then regarding harvest, which was done either using plot harvest or combine harvest, almost all the states used combine harvest, except North Carolina, which did hand harvest in all 3 years, and Kentucky hand harvested corn in their final year. And that harvesting occurred in December in New York and Pennsylvania. Other than that, it was either mid-September, October, and this was true in all 3 years as well. All right, now let's move on to our results. And I've divided our results into two parts, one about cover crop, and next would be about yield, corn yield, and then optimum nitrogen rates. This is a busy slide. I'll walk you through this. So we have 3 variables here. This is summary of biomass, nitrogen uptake, which is nitrogen that's been taken up in the biomass, which is in kilogram nitrogen per hectare. And then there is carbon to nitrogen ratio. And this is for 4 different cover crop treatments, which are represented through different colors. The first one is fallow, second legume, third mixture, and then rye. And different numbers represent average values for these variables. And then the letters are significant letters. So when you compare the significant letters, we're not comparing this lowercase a with uppercase A with italics. It is compared within this vertical stacks. So what we found was mixture and rye had highest biomass and legume had lowest compared to the other two. And legume and mixture had highest nitrogen uptake in their biomass, which they were not different from each other, but rye had lowest nitrogen uptake in their biomass as compared to legume and mixture. In case of carbon to nitrogen ratio, legume had lowest, rye had highest, and mixture was an intermediate between legume and rye. Now you might be wondering, like, because these are treatments and we had multiple species within the treatments, right? Because where is this number for fallow coming from? It's weed species. And for legume, it is weed as well as legume. For mixture, it's rye, legume, as well as weed. And for rye, it's the same case, rye and weed. So what is the proportion? What is the contribution of rye in mixture as compared to legume, right? So wait for that. And these here shows the proportion of the species in the cover crop treatments. And here we are looking at two figures. One, this whole thing, this is weight, that is by biomass, and this is nitrogen uptake. And here, this pink color represents legume, and this blue is always representing weed. Green is always representing rye. So around 75% of the legume by biomass is actually by legume, contribution by legume, and the rest around 25% is weed. For rye, around 95% is of that biomass is the rye itself, and around 5% is weed. So it's pretty evident that weed infestation is higher in legume cover crop treatment. In case of mixture, there is a higher proportion of rye than legume by weight as well as by nitrogen uptake. I also looked at correlation between the biomass and nitrogen uptake, and they were all positively for our cover crop treatments, and they were all positively correlated with strong positive correlation of legume biomass with their nitrogen uptake in their biomass. And we found that for every 1,000 kilogram increase in the biomass, nitrogen uptake increased by 24 kilogram per hectare in legume, 16 kilogram in mixture, and 12 in rye cover crop treatments. And although we did see that there was overall highest nitrogen uptake in our legume as well as mixture, because they were not statistically different. However, much of the legume nitrogen was actually derived from atmosphere rather than soil. So we'll look at this table and it will be pretty clear. So this is the average nitrogen uptake, which the number that we just saw. So for legume, if it was 69 kilogram of the nitrogen was taken up in their biomass, 37 kilogram was from fixation. So this is, although 37 and 32 are not very different from each other, but more than 50% of that is fixed from atmosphere. And then in mixture, out of 74, this 21 kilogram is the contribution of legume, because this is the nitrogen that's been fixed from the atmosphere. And in this 53, this is the nitrogen taken up from soil by rye as well as legume in mixture. So legume, either in monoculture or in mixture, tend to fix nitrogen from the atmosphere more than the nitrogen retained from the soil. Sorry, there's a message in chat. We'll figure that out later. So you might be wondering about the species. So there are two different species in legume, that is clover and vetch, as I mentioned before. So the percentage of the nitrogen that was derived from fixation was greater for clover overall than for hairy vetch in mixture, as well as either in legume monoculture or in mixture. And these plot, they determine the percentage of nitrogen derived from fixation, and this is similar, just the biomass. This is the nitrogen derived from fixation, but in kilogram nitrogen per hectare. . 。 . 。 。 . 。 . 。 . 。 . 。 . 。 . 。 . 。 . 。 . 。 . 。 . 。 . 。 . 。 . 。 . 。 . 。 . 。 . 。 . 。 . 。 . 。 . 。 .
.
So looking at the correlation between legume biomass and nitrogen derived from fixation, again, positively correlated for both legume monoculture and mixture. And these two different shapes represent clover and vetch. And what we found was for every calculating the slope, what we found that for every 1,000 kilogram increase in the legume biomass, the legume cover crop fixes an additional of 16 kilogram nitrogen per hectare. All right, so this leads us to our second part of the result, which is about yield and about optimum nitrogen rates. So our average grain yield averaged across years and cover crop treatments ranged from 7.9 to 13.4 megagram per hectare. And we had total, don't be alarmed with this slide. I know it's a busy slide, but this is just showing you the original data just averaged across replication. And the data is yield megagram per hectare across nitrogen rates. And we have 12 sites, 3 years, so 36 site years. I haven't done anything with this. This is, like I said, original yield data for all our cover crop treatments. And just wanted to see, because now we want to use this in order to fit in our quadratic plateau curve so that we can get optimum nitrogen rate following all our cover crop treatments, right? So for that, just wanted to see like how the data looks. And what I have highlighted here, or something that we wanted to talk about, is that for Maryland second year, it looks like they are sort of showing this linear increase in their yield with an input. And for Vermont, all 3 years, it kind of looks like there is no response, no yield response to nitrogen fertilization. Similar case for Wisconsin, second and third year. But in first year, for Wisconsin, it looks like there is a slight, there's a slight quadratic response, maybe a plateau as well, for probably one of the cover crop treatments. But this is just the visual interpretation. So we wanted to do some testing as well. So what we did was I tested this for all of those 36 site years, but presented these figures are those 6 site years, which did not show a significant quadratic, which had non-significant quadratic term for quadratic curves. So we fit it in linear mixed effect model with grain yield as our response variable, and the fixed effects were due to cover, nitrogen rate, location years, and random effects were due to location years and replications. So if we had even one of the cover crops showed a significant quadratic term, then I have used that in the analysis. And that's what I did for some of the site years, like Wisconsin first year. But unfortunately, for these site years, we're not able to use only this site here in the analysis because they had a non-significant quadratic terms. So the total site here in analysis, by analysis, I mean for the optimum end rate analysis, not the previous results that I showed. So total site here in analysis is 30. So we still have all sites except for, unfortunately, Vermont. So we have Maryland's first and third year data here, Wisconsin first year. So we were able to use these in order to do our further analysis now. Here's the result. So the numbers here show agronomic optimum nitrogen rates, where there is 253 kilogram of nitrogen, kilogram nitrogen per hectare following fallow, 254 following legume, and they were not different from each other. A mixture and rye had higher agronomic optimum nitrogen rate, but they were not different from each other, whereas legume was significantly, agronomic optimum nitrogen rate was significantly lower for legume as compared to mixture and rye. And this black dashed line shows average agronomic optimum nitrogen rate, and orange dashed line shows economic optimum nitrogen rate, which was 219 kilogram nitrogen per hectare. And average agronomic was 243 kilogram nitrogen per hectare. And this is averaged across treatments. And economic optimum nitrogen rate for all the treatments is over here, where legume was lowest 221, and mixture was 274, and rye was 293. So we saw that for agronomic or economic, there were optimum nitrogen rates were lowest for legume and highest for mixture and rye. At under no fertilizer application, we saw that rye had so these are the yield at no zero nitrogen fertilizer application. That is no nitrogen fertilizer. And here we saw that yield at 0, and legume had highest yield, rye had lowest, and mixture was an intermediate between legume and rye. And still legume had higher yield than fallow, and whereas rye and mixture were lower than fallow. And when we look at that in percentage, around 32%, rye exhibited around 32% lower yield than fallow at no nitrogen, zero nitrogen. And whereas a mixture was 14% lower than fallow, and legume was 9% higher than fallow. But at optimum nitrogen rate, so at agronomic optimum nitrogen rate, yield was not different from each other. So yield following all of the cover crops were not different from each other. It kind of ranged from 12.5 to 13, but they were not different from each other. They're suggesting that our yield penalties, they may be overcome by adding adequate nitrogen fertilizer. And this was same case when at economic optimum nitrogen rate. So these are yield at economic optimum nitrogen rate following all the cover crops, and they were not different from each other, but it ranged from, again, 12.4 to 13. Now, after all of that, I also did some economic analysis in order to see the net benefit associated with all these treatments. So just a heads up that so far I've shown all the data in SI unit, but now I'm going to use American unit like bushel per acre, pounds nitrogen. Previously, I was talking about megagram per hectare, kilogram nitrogen. Just for easy interpretation and to get better perspective. So now we already have economic optimum nitrogen rate, and I showed that data, and we have yield at economic optimum nitrogen rate, and this is in bushel per acre. And and economic optimum nitrogen rates are pounds of nitrogen per acre. So what we want, in order to get a net benefit, we wanted to calculate the revenue that has been gained from yield gain, which was negative because yield was higher at fallow as compared to legume mixture and rye. So legume mixture and rye had lower yield as compared to fallow. Hence, the difference was negative. Therefore, there was no positive net gain, revenue gain from yield gain. However, for optimum nitrogen rate, legume exhibited lower economic optimum nitrogen rate compared to fallow. Therefore, there was a slight revenue gain from nitrogen fertilizer saving. And the cost that we used for fertilizer was 60 cents. So 60 cents per pounds of nitrogen. And for corn price, it was $4.60 per bushel. And then seeding rate, we also incorporate. So this is our cost, which includes our total cost, includes the cost of seed, which is higher for legume, and we average this cost for hairy vetch and clover together over here. And to get the cost of seed for mixture, we definitely use the seeding rate that was used, seeding rate of legume and rye that was used in mixture, which was slightly lower than legume in monoculture and rye in monoculture. And then this total cost also includes the cost of planting and termination, which was about $30 is what I added on top of like seed cost. And all this cost is we got it from Midwestern Cover Crop Council website. So after that, the net negative is not positive. So there's not a positive dollar gain following cover crops, but after when nitrogen was applied at UNR, that's the case, but legume was still the most economical treatment as compared to mixture and rye because of its slight gain in nitrogen fertilizer saving. This leads me to my conclusions, like depending on different cover crop species, as we saw, optimum nitrogen rates for corn also varies. And cover crop mixture had similar biomass production as that of rye, and which were high, and similar nitrogen uptake as legume, which was high, and intermediate carbon to nitrogen ratio. So carbon to nitrogen ratio for legume was high, lowest, rye was high, and mixture was an intermediate. And there was higher proportion of rye than legume in mixture, and that was true by biomass as well as by nitrogen uptake. And most of the legume nitrogen, either grown in monoculture or mixture, they are derived from atmosphere, then soil. Agronomic as well as economic, both optimum nitrogen rates were lowest for legume and highest for rye, and it was true for mixture as well. And in the absence of nitrogen fertilizer, legume had highest, while rye had lowest yield. And at optimum nitrogen rate, there was no yield differences among cover crops. And when nitrogen was applied at economic optimum nitrogen rate, cover crops did not have any yield benefit, while legume showed a minor benefit in fertilizer saving. The added cost of seeds resulted in a net negative economic benefit. This leads to the end of my slide, but I really want to end it by acknowledging important people here. Dr. Hannah Poffenberger, my advisor, I really want to thank her for immense, advice, support during this the whole project, as well as this presentation. She also contributed to some of the slides and has great advice and is really available all the time. And I want to thank Lucas, who was involved in this project more like deeply than I was, because he was a previous postdoc for, I think, the last couple of years before I started in last September. So he did lots of the work, and I was just crunching numbers. And I really want to thank and special thanks to all of you. If your name is missing from here, I'm really sorry. This is a genuine mistake. But I want to thank you for doing the real work. You were in the field, collected data, did all data cleaning, sending it to, uploading it on the website and everything. So thank you so much. And also sorry for sending you so many emails, but thank you for responding to them. And my last but not the least, I want to thank our funder, USDA PSA project. Thank you all, and I look forward to your questions. Thanks.
Ashmita, thank you so much for this presentation. It was a lot of number crunching from a lot of different site years and a lot of different places. So thank you. I have the privilege, I think, as host of asking a question. There's a few in the chat also that that I'd like us to look at in a second. But I'm wondering if you can that take-home message around the cost of using cover crops, that bottom line cost. I'm wondering if you have thoughts about what the messaging might be then to growers about nevertheless the benefits, or how we should be thinking about talking with growers about the fact that it looks like from an economic perspective they're still going to lose money in this process.
It's sad to see that we're going to lose money, but still, I think there's legume is more economically beneficial compared to the other cover crops, but with growers, I would say we need more research. We need to know, like, if we could, and I've also if we could if there's a way to increase yield at economic optimum nitrogen rate, because if we could get this increase this revenue gain from yield, then with the positive, with the fertilizer nitrogen revenue saved in nitrogen fertilizer or nitrogen fertilizer cost cutting, we should be able to decrease this negative and increase our positive benefit associated with legume cover crops. So that's that. But I think I don't know. I think other people will be able to provide more perspective to this more than I do working with growers. But yeah, I don't want to tell them that, yeah, you're losing $83 per acre and still you should do this. But yeah, there are environmental costs associated with it for sure. But then, yeah.
Yeah. Well, I asked that because we've heard over and over about from growers not to think about, not to be thinking about yield as the only bottom line, because if they're, putting in less money, then, they can still make up the cost. So anyway, it's one of those things about how do we think about the messaging of this, of these kinds of findings. So there are a couple of comments and questions in the chat. Michael asks, and followed by Dean, how do you determine measure BNF for legumes? And Dean says, how could you tell that nitrogen came from fixation versus soil?
I think I would let Hannah talk about this. I think she would be able to speak to it better than I do, because the nitrogen fixation slide I stole from her, although I can speak to that as well, but I think she would be able to provide better insight to it. We have like N15 is what we were able to analyze, so and then that's how we calculated nitrogen from fixation versus from soil.
Yes, we analyzed so we separated the cover crop samples by species. So we had legume, rye, and weeds separated for each of the treatments. And then we analyzed the biomass samples for the N15, just using a stable isotope N15 values, and then used the rye and monoculture as a reference for nitrogen derived from the soil. And then we use a B value to account for any kind of offset associated with nitrogen coming from the atmosphere. So they recommend, rather than using a delta value of 0 for nitrogen from the atmosphere, to use like a slightly negative value to account for the fact that the plants move N15 in their shoots versus roots differently. So that's probably more detail that you wanted to know, but basically the stable isotope N15 method.
There are quite a few questions in the chat, but I want to invite those folks who threw them in the chat to just jump on and ask your question. If not, I can read them, but it's better to hear from you.
I'll jump in. Yeah, question I had is regarding, so we see these different responses with the different cover crops. And I'm wondering if the same emerged population, corn population, was achieved across the different cover crop, treatments. And I guess why I'm thinking about that is, one of the things that is cited often in terms of no-till planting into cover crops is, planter issues and having lower emerged stands. I'm wondering if that is a way of thinking about potential yield reductions as well. Thanks.
I'm so sorry. Could you repeat the last part of your question?
Yeah. So each site used the same seeding rate for corn into the no-till planting corn into the cover crops, and I'm wondering, Ashmita, if what were the emerged corn populations, just before harvest? For example, did we experience higher plant populations in the fallow compared to the cover cropped treatments?
That's a great point. That's something we definitely need to look into, differentiate that by corn population. It was almost similar, though. I did not see a very significant differences across in terms of the emerged corn population before harvest. But yeah, in terms of cover crop, that emerging issue and was seen for legume in some cases, but I don't think that was different for corn population though. Is there anything else that you might have noticed, Hannah?
I don't think that we've really analyzed the corn populations yet.
I have
So
reported, but
what
not,
was that?
yeah, I have reported them in my sheet, but not analyzed them yet. But I don't see like significant differences, like, nothing stood out.
We did, yeah, I will say like Ashmita did a really meticulous job of like checking any outlying yield values. And part of that was like, okay, I mean, that I think we did dig into the population just to make sure that a low yield was not being explained by just a lack of plants in the plot. So any like really extremely low populations we looked into, but I don't think we really had any. We were looking out for that, but I don't think we had any cases where an outlier was caused by low population. But in terms of just like analyzing the average effects of cover crops on population, we haven't really done that yet, but we should.
I have a question
Thanks.
on your economic analysis. I'm assuming on your slide where it says like minus $83 per acre or so, those are averages over all your fields. Did you, or maybe you noticed, or maybe, or was there any trend for the same field if that cost decrease or not? Do we expect that would happen and potentially become marginal when if there is a carbon credit market out there that can, superimpose that cost? Or what are your thoughts?
Again, great point. I haven't looked at like carbon credits or did that sort of analysis yet, but yeah, this has not been looked at just the particular field level, so not done for like 36 different sites. This is the average yield, yield at economic opportunity following all the treatments, and then that's how we were able to analyze it. But yeah, I haven't done that in short to answer your question.
Yeah, good presentation. Thank you.
Thanks.
And also,
kind
just
of
to add on
tricky.
that,
Sorry, go.
yeah, I think just to add on that, Jones, I think this field each year, although this was connected for 3 years, those are connected on 3 different fields. So not necessarily having the same cover crop treatment over multiple years. So it's like a one single year effect of cover crops.
Yeah, and yeah, I was going to make the same point because with the longer time frame, maybe the yield penalties would go away and decreasing or increasing fertilizer requirements would also go away, and then we see carbon buildup happening. So this is just kind of a short-term economic budget.
Jody asked a sort of similar question or a related question, thinking about the broader ecosystem services, but I think we're looking at a similar answer, right? In terms of the short-term versus the long-term benefits. And this makes me think that maybe, Chris is asking about region by region differences. Oh, no, sorry. Someone else was asking about region by region differences, but Chris was asking about the seeding rate. Chris, do you want to jump in?
Sure. Yeah, I was just gonna, I think as experimentalists, we always have to, we have these debates about like how to treat seeding rates. And, I think it was a lot of good reasons to choose the same seeding rate across all the locations. But there is an artificiality about that. for the economics, because we just know that they're like in our southern locations, we would never use that much seed here. And we would probably get the exact same nitrogen return to the system from much lower rate. So like, you know, is there a way to address that in the write-up? But I would overall, first of all, this was phenomenal stuff. I love the way you've glued all this together. These are such a big jobs to do this. And so it really, these types of analysis invite us to do the economic returns. I've just always struggled with the parts of that. But I'm also really excited about showing a negative. I mean, I think sometimes our community has been shy about that. My argument has been, I think there's enough evidence pointing out in general, we're not, that there needs to be a subsidy for cover cropping. And we're trying to achieve all these environmental externalities, and we shouldn't shy away from saying, no, this is, you know, establishing the correct subsidy rate would be one outcome of this type of thing that is really important.
Yeah, I would like to continue on that one, Chris. So I agree with that, because a lot of times we're helping people starting doing cover crops, but we need to also help people and give them incentives to continue. And I mean like long-term continue. And I mean like, you know, if we're talking about the needs for climate mitigation, erosion control, and soil health in our country, I'm talking like 20 years to help farmers continue down this road with keeping our soils productive for our country. So this isn't just, you know, 3 to 5 years and you're done, you should get the hang of it. No, we need to be able to support farmers for long term and have peer-to-peer help for them and help them understand in the beginning it will be challenging. It will probably, you know, give a bite economically, but our country's here to support this kind of farming. Thank you.
Thanks, Jody. Yeah, and Dean, it was Dean who was asking about whether they were more profitable in some regions but not others. And I think this kind of comes back to, again, getting to this economic bottom line in some ways too, and thinking about how that should be informing or shaping policy. There's this question of, okay, like where do the incentives levels need to be set in different places to recognize the varying cost of these practices? Did you see differences region by region?
Well, that's something we haven't done yet. So we, due to lots of variations in the data set itself, we're not able to fit in our curves and get economic optimum nitrogen rates or AONR for reasons specific. And that's something that we are still, it's on the back of our mind. And because in order to get that economics, I need to have AONR and then yield at AONR values for, if you're talking about just the reason specific. And that's something we have been talking with our group, like back and forth. And yeah, we're definitely, we would like to see that as well. Even for optimum nitrogen rates, we would like to see it for particular reasons. And then do economics for the afterwards as well. But that's something, yeah, we are still, it's still not done.
Yeah, I think the translation of this kind of work into policy is just super important. So thank you very much for it.
Yeah, thanks for bringing that up.
I feel like the legumes didn't grow too well in our northern sites, but so I just feel like if we looked at it in the southern sites, apart from Chris's point that the seeding rates would be lower, I think we just get a lot more benefit from the legumes in terms of savings in nitrogen fertilizer and possibly more yield benefits because they're growing bigger and fixing more nitrogen. So I think doing it by region, we may see more positives, at least for where the legumes grow well.
Any other questions or any that I missed in the chat? Well, Ashmita, I don't think that I've seen as vibrant a chat thread for any of the PSA seminars. So I think that you really stimulated a lot of thinking and a lot of discussion today. So thank you so very much. This is really important work. And as I said, I think it really informs how we need to think about policy going forward, how we need to think about supporting growers, and really glad that you're doing all this work. Thank you so much. Thank you to everyone who joined us today. We will be off in June, July, and August, but we'll be back in September for Carlene and her crew talking about CE2. So thank you all very, very much. And we'll see you here in September. Take care, y'all.
Thank you, Jen. And thank you all for giving me this platform and opportunity. Special thanks to our PSA group, Hannah, everybody. Yeah, thank you. And this was really good, like putting this presentation together. I also learned a lot and did the economics for the first time. So it was super interesting. Yeah, good to see our work coming together. Yes, I'll definitely share my link to the paper once it's ready and published, Danielle. Thanks all.